Hey y'all, Brown's back. This time we're doing some more histology and we're getting into reproductive histology. So today we are going to be investigating, as you might guess from the cover of this video, the testes. The testes are the primary reproductive organ in men. And when we say primary reproductive organ, what we mean is the organ where gametes or sex cells are made. These are the cells that unite to form a new person. And in men, these form in the organ called the testes. Testes being plural, the singular being testis, T-E-S-T-I-S, will be the singular form, like so. And the testes contain these little compartments full of tightly coiled tubules called the seminiferous tubules, which is what we're seeing over here on the left-hand side of the screen in cross-section. So what I want to do now is kind of zoom in on a section of testis and look at kind of the general histology of the testes. As I mentioned previously, germ cell production, that is, in the case of men, sperm cell production, takes place inside these tightly coiled tubules called the seminiferous tubules. And as you know, if you cut a tube in cross-section, you get a circle. So as I go in here, I can draw a circle around a single seminiferous tubule. Very nice. You can see if you look closely that the seminiferous tubule is surrounded by a thin layer of regular connective tissue. And since this is regular connective tissue, you're going to see numerous fibroblasts. There's a couple out here in the edge, about 1 o'clock or so if we're looking at this as a clock face. But I want to draw your attention to the more numerous cells with their very highly compressed nuclei. So here we can see this very, very flattened nucleus. There's one down here at the bottom as well. These cells over here are called myoid cells or myoid epithelial cells. And these myoid cells are weakly contractile. They're going to act kind of like smooth muscle, and they're going to gently compress the seminiferous tubule to move the fluid and the cells that are contained in the fluid out here in the lumen along into the genital ducts. Inside this connective tissue capsule, we find the layer of tissue that is going to become the sperm cells, and this is called the germinal epithelium. Germinal because we're making germ cells, and epithelium because duh. We're going to look at the germinal epithelium in a little bit more detail here very shortly. In the next image, we're going to look more carefully at how we make those germ cells. But before we do that, I want to talk about one other part of what we're looking at here, and that is the interstitium, the area in between the seminiferous tubules. If you look in the bottom left-hand corner of your screen, you'll see a little pack of cells, these nice broad, round, spherical nuclei cells. These are called oh goodness, that was cool. I don't know how I did that, but that was neat. These are called interstitial cells. Let's just make that connect. Very good. When I was actually a young man and was learning this stuff for the first time, they were called the cells of Leydig, but now we simply call them interstitial cells. And these interstitial cells function to secrete testosterone. These are the cells that are responsible. I misspelled testosterone. Let's fix this. Whee! That's what erasers are for. Testosterone. Very good. These are the cells that are responsible for the reason that men don't look like boys. It's the testosterone. And these interstitial cells are stimulated by the hormone called interstitial cell stimulating hormone, or ICSH, which is the exact same molecule as luteinizing hormone in women. So it's going to stimulate these cells. They're going to produce testosterone. These cells are initially active during the fetal stage of life, and it is in fact these cells and the testosterone that they produce that are responsible for the developing fetus taking on kind of the male body plan. The default body plan for humans is female. The male genital development will only occur if testosterone is present. And so if testosterone is not present, even if somebody has a Y chromosome, or if somebody doesn't have the testosterone receptor, then they won't develop male genitalia. So it's really fascinating if you ever want to learn more about androgen insensitivity syndrome, those are people who are XY but don't see testosterone, so they develop as developmentally as females. It's really neat stuff. But in vast majority of folks that have a Y chromosome, these cells are active up through about the 4th month of development, and then they shut off and they stay off until the little boy goes into puberty, sometime around 9-10 years old. So let's look more closely at the germinal epithelia and some of the cells there that are actually going to go into making germ cells or sperm cells. So we've zoomed in a little bit here. We can see a little better the cells of the germinal epithelium. We can see our myoid cell out here. This is very nice. You can see a weakly standing fibroblast nucleus there. But we're interested in the cells of the germinal epithelium. And the first cell I want to draw your attention to are these guys right out here. Here's an example of one. This is called a spermatogonium. And the spermatogonia are kind of like little spermy stem cells. A man of healthy reproductive age is producing sperm to the tune of hundreds of millions a day. And that's a lot of cells to be making. So if we're going to develop cells into sperm cells, we need to replace the ones that got turned into sperm. So these spermatogonia are constantly undergoing mitosis in order to replenish the cells that become sperm cells. Now, there are actually two different kinds of spermatogonia. You can't tell the difference between them histologically, so we're not going to learn how to tell them apart or anything, because you can't. But there's one called the type A spermatogonium that's a little bit less differentiated. It's much more stem cell-y, and it's just doing mitosis. And then some of those can become type B spermatogonia, which are cells that have now terminally differentiated and are on the track to becoming sperm cells. That's all we're going to get into about that. We don't need to do any more. As we move further in towards the lumen of the seminiferous tubule, we see some much larger cells, like this guy right here, like this guy right here. These cells are called spermatocytes. And these are the cells that are in the process of meiosis. I can't spell today. You're welcome for the entertainment. There we go. Meiosis. Very good. And there's actually two different kinds of spermatocytes. We have primary spermatocytes, which are in the process of meiosis 1, and we have secondary spermatocytes, which are in the process of meiosis 2. It's really hard to tell them apart histologically, especially kind of at this introductory level. But if you guess primary spermatocyte, 9 times out of 10 you're going to be right, because meiosis 1 freezes in prophase 1 for about 21 days, or 22 days, I think. Yeah, we'll say 22 days. It's about 3 weeks. So there's a good chance that any spermatocyte you see is probably a primary spermatocyte, because they stay frozen in prophase 1 for 3 weeks and then very rapidly move through the rest of meiosis 1 and all of meiosis 2 until they become one of these little guys, and that's a spermatid. So a spermatid is all done with meiosis. It's a haploid cell, so it's going to have 23 chromosomes, each with just one chromatid. So all done with meiosis, but it still has some morphological development to undergo. A spermatid is not capable of fertilizing an egg. It has to be a spermatozoan. Before we get to the full development of the spermatid and the spermatozoan, however, we need to look at one more cell that's present in the germinal epithelium. These cells are called the spermatogenic line. And most of the cells you look at when you look at a seminiferous tubule in section are cells of the spermatogenic line. But there's still one other cell that's very important and necessary for the production of the germinal line here, and that is the Sustentacular or Sertoli cells. On the right, we have the image we were just looking at, and what I'm going to do is draw kind of an outline around a single Sustentacular cell. I'm going to do my best to. Oops. Very good. Come around this way. And like so. And like so. So, as best as I can tell, everything inside this black line is a single sustentacular cell. The nucleus is here. You can tell this is a sustentacular or a Sertoli, that's kind of the old name for it, cell, because it's got a lot of euchromatin in there and a pretty prominent nucleolus, whereas these cells that are in the process of meiosis have a lot more condensed chromatin in them. Sertoli cells are broadly pyramidal, generally tend to be broader towards the outside edge of the seminiferous tubule and more narrow as you get towards the lumen. And their primary function, as the name Sustentacular would suggest, is to support, to sustain the developing spermatocytes and spermatids. As those spermatocytes and spermatids are developing, especially as the spermatids are developing from early spermatids to late spermatids, which I'll get into more in just a second, they're going to be shedding cytoplasm. They're going to be getting rid of anything that's not needed to fertilize an egg cell. So as those developing spermatocytes and especially the spermatids are shedding cytoplasm, it is the sustentacular or Sertoli cells that will endocytose that shed cytoplasm. These guys are also secreting testicular fluid, so they are secreting the fluid that fills the lumen and provides a transport medium for the fully mature spermatozoa. As you can see in the image on the left, they're also carrying out a very important function. If you look at that number 8 and follow the arrow down, you'll notice that these two Sertoli cells, number 7 and number 7 here, are attached to one another via a tight junction. Right there, there's a tight junction. Which means that everything on the other side of this dashed line, everything above the dashed line that I just drew, is separated from the external environment here. So this Sertoli cell is connected to, the nucleus is down there, but we'll just say there's another one right over here. And these guys are going to be connected to one another by these tight junctions. And that's going to form what's known as the blood-testis barrier. This is probably something you've never thought about before, but let's look at it just a little more closely. This cell down here with the number 2, so 1 is the basement membrane, number 2 is the cell. This is a spermatogonium. These are stem cells. These are genetically identical to every other cell in this man's body, which means that his immune system sees these as self. However, once the type B spermatogonia enter into prophase 1 of meiosis and become spermatocytes, primary spermatocytes, they're going to engage in genetic recombination. Crossing over is going to be occurring during that 3 weeks it's frozen in prophase 1, which means that every cell on the other side of this dashed line is now non-self and subject to immune attack. For this reason, the sustentacular cells, and specifically these tight junctions, are going to shield the developing spermatocytes and spermatids, and eventually the spermatozoa, which are genetically different from the man who created them, shield them from immune attack. So that's pretty cool. The last thing we need to talk about before we end our video, and we're already 15 minutes in, is the development of early spermatids. So this little guy right here is an early spermatid into one of these little guys right here, which is a late spermatid. And then from one of these little guys right here, the late spermatid, into a fully mature sperm cell. This process is called spermiogenesis. Meiosis, going from spermatogonium to spermatocyte to spermatid, is called spermatogenesis, but going from early spermatid to late spermatid to spermatozoan is a purely morphological change and is called spermiogenesis. Some of the major features of spermiogenesis that I want you to pay attention to are: 1, the nucleus is going to compress. Ooh, bad handwriting. 2, we're going to lose lots of cytoplasm. So cytoplasm Be gone. It's going to grow a tail. See it right there? So it's going to grow this long tail. And it's also going to develop a little pouch at the front called an acrosome. So all of these things occur during the process of spermiogenesis. If we look at this lovely cartoon of a mature sperm cell by Wikimedia Commons user Lady of Hats, this is a really nice work, and she did this totally for free. She put this thing into the public domain, which is really fantastic. We're going to see that the sperm broadly can be constructed of a head and a tail. The head is going to contain the nucleus. I often use the analogy that a spermatozoan is kind of like a little guided missile. It's a little guided missile of DNA that is seeking out its target, the egg cell. The nucleus is the payload. That's the thing it has to deliver inside the cytoplasm of the egg cell so that we can get syngamy and eventually a zygote. But it's not easy. That egg cell is well protected by a thick outer covering called the zona pellucida that the sperm cell is going to have to burrow through in order to fertilize the egg. Fortunately, the acrosome, which we saw down here, contains enzymes that will help break down that zona pellucida, allowing the sperm cell to penetrate the egg cell. We also notice that the midpiece here that kind of connects the head to the rest of the tail contains numerous mitochondria. These are the smallest cells that a human can make. They are tiny, and they have to swim a really long way. The mitochondria are going to break down sugars, turn it into ATP to power the flagellum that makes up most of the tail. This flagellum will beat much like a propeller, moving the sperm cell through the female reproductive tract and allowing it to eventually find an egg cell, fertilize it, and make a new person. Well, that's it for now. Sorry the video went a little bit long. This is a really fascinating topic, but I hope you enjoyed it. Have fun, study hard. We'll see you later.